Good morning, everyone. Hello. Thank you for being here in person and virtually. It is such a great honor and pleasure to be hosting Dr. Paul Marker from the University of Wisconsin in Madison. So Paul completed his undergrad degree at Grinnell College, and then he went to graduate school at Stanford in developmental biology. Correct me if I'm wrong. And then he postdoced at UCSF in Jerry Cunha's lab, and Jerry Cunha was quite a well-known cancer researcher who innovated the ability to grow cells in the kidney capsule, which created a microenvironment. And then Paul got his first position here at the University of Minnesota as an assistant professor, and we were so lucky to get him here, and his wife, Dr. Collier. And Paul was just a fabulous, fabulous faculty. His office was right next to mine, and Paul he's everything I'm not. He's very measured and thoughtful. And so whenever I had a problem, I would go talk to Paul, and he would always have this wisdom, and this very now, Carol, you got to do it this way. You can't do it that way. And so I learned a lot of thoughtful thinking, and his wisdom really helped me a lot. And then tragically, he was recruited away to Madison, tragically for us, but where he rose very rapidly through the ranks. He became the associate dean for research of the School of Pharmacy. He became the vice chair of his division, and now he serves as the chair of his division, which in the School of Pharmacy is really a department chair. And he remains very active in the field of prostate development, prostate diseases, including prostate cancer. And more recently, it has entered the field of breast cancer. And he uses quite elegantly mouse models, as well as biochemistry, to demonstrate mechanisms of development, disease processes, and cancer. And so happy to have him here as our first kickoff seminar. And again, thank you for coming, and I hope you enjoy Paul's talk. Thanks very much.
Well, thank you, Carol, for the kind introduction. It's great to be here. Meet some new people, see people I've known for a long time. And I got to say, the last 2.5 years have been strange, right? This is my first in-person seminar that I've come to deliver since the COVID pandemic. Today, I want to talk to you about the role of growth hormone in the IGF-1 axis in both breast and prostate cancer. It'll be mostly about prostate cancer because really that's what I'm trained in. That's what I mainly do. But you'll see why there was a diversion into some models for breast cancer as well. So in terms of prostate cancer, this image on the right is a bisected human prostate gland. And this is a branched tubuloalveolar gland, which is about the size of a walnut. Even though it's pretty small, it's important. It makes about 30% of the seminal fluid. And if you look at the estimates from the American Cancer Society, in 2022, they expect there'll be almost 270,000 cases of prostate cancer in the United States. And they expect there'll be over 34,000 deaths from prostate cancer in the United States, which makes it the second leading cause of male cancer death after lung cancer. Now, benign prostatic hyperplasia, which is a benign enlargement of the gland, is also medically really important. It happens in over 50% of men over 60 years of age. It causes lower urinary tract symptoms. I'm part of a U54 grant that studies this, but because I'm here for the Cancer Center seminar, I'm not going to tell you about any of the research that we're doing on BPH today. But I will acknowledge the people I work with at the end of the talk. Now, in terms of, again, prostate cancer background, the lifetime risk in the US is thought to be out 1 in 6 men get prostate cancer. And it does vary by ethnicity. And there's a chart here I've put on the right-hand side from the NCI that shows the incidence per 100,000 men. The incidence does vary with ethnicity, but as you dig deeper into this with epidemiology. There's a likely role for diet. So, for example, the rates are very low for Asian and Pacific Islanders. Epidemiologists have looked and they followed individuals from those cultures that have moved to the US. And if they move to the US and they maintain their diet and traditional lifestyle, they keep their low rate of prostate cancer. If they adopt a more Western diet, US lifestyle, their rates of prostate cancer go up dramatically. So, thinking about mammals, dogs are the only other species that really get prostate cancer just by living. And that's been studied some. Many of us love to study mice because mice are convenient as a laboratory animal. And it is also the case that if you engineer the specific mutations that are found in human prostate cancers into mice, they get prostate cancer too. And that'll be relevant because I'll tell you about a couple of different animal mouse models that we've been studying of prostate cancer. Now, in terms of features of prostate cancer progression, so this is a cartoon adapted from the article that I cite at the bottom of this slide, but normally this is a secretory gland, and you have this layer of epithelial cells that secretes proteins and other materials needed for reproductive function. With mutations, those cells can pile up within the gland. You get what's called prostatic intraepithelial neoplasia, or PIN. That's an early lesion of prostate cancer. It's a precursor because it's not cancerous, but if you look, there are mutations that are characteristic of prostate cancer in those cells. Eventually, you can get invasive cancer where the cells break through the basement membrane, invade surrounding tissues. This is also a cancer that metastasizes. And of course, the metastatic cancer is what's lethal. I'm not going to talk extensively about it, but there are a number of therapies for prostate cancer, including hormone ablation. It's a very androgen-dependent disease, but most men with advanced disease get treated with androgen ablation. It progresses to castration-resistant prostate cancer. That is lethal. There's minimal things you can do that extend life a little bit, but really we need new pathways to target in this disease to address these lethal cases of castration-resistant prostate cancer. And one of the things I'm going to tell you about today is the potential role of the growth hormone IGF-1 axis as a target. And one of the reasons we think this might be important is the absence of prostate cancers in patients with so-called Lamone Syndrome. So I'm going to tell you a little bit, and that's a syndrome where people have inactive growth hormone signaling. So just to give you an overview of how growth hormone signaling works. So normally the anterior pituitary, which is at the lower end of the brain, secretes growth hormone. And one of the major target tissues for growth hormone signaling is the liver. So the liver expresses a high amount of growth hormone receptor. And we know that one of the major outcomes of growth hormone signaling in the liver is secretion of IGF-1. And data suggests that about 90% of the circulating IGF-1 is coming from the liver and due to growth hormone stimulation. IGF-1 in turn is an endocrine hormone. It circulates throughout the body and it binds to receptors in many different tissues, including the mammary gland and the prostate. And it is in part mediating the effects of growth hormone in those target tissues. Now, there's another potential aspect of growth hormone signaling, which is that the liver isn't the only tissue that expresses the growth hormone receptor. Many other tissues also express growth hormone receptor. This includes cells within the mammary gland, cells within the prostate. And as we're conducting our studies, one of our questions is, there could be potentially indirect effects on prostate cancers of the actions of growth hormone. There could also be direct effects because growth hormone receptors are expressed by prostate epithelial cells. I'll talk mostly about prostate cancer today, but I'll talk a little bit about breast cancer. They're also expressed by epithelial cells in the mammary gland. So I alluded to Lorone Syndrome patients. So what are these patients? So now this is presenting work of others, but this photograph includes image of a number of people who have so-called Lorone Syndrome. This is a form of dwarfism, and these individuals have a non-functional growth hormone receptor. And overall, they're largely healthy. This particular population has been followed, where they followed in the study that I cite here in this slide, 143 individuals with growth hormone receptor deficiency, and then over 1,600 of their unaffected relatives. And they followed them for basically mortality and disease in all forms. And what they found is that although about 20% of the relatives with intact growth hormone signaling died of cancer, which is basically the expected rate based on the population, none of the growth hormone receptor deficient individuals died of cancer. And relevant to my talk, there was a 0% incidence of either breast or prostate cancer amongst the Lorone syndrome patients. So this kind of implicated the growth hormone IGF-1 axis as being at least permissive for prostate cancer and breast cancer. Now, there is a mouse model for this. So in this image on the right, the mouse on the left is a wild type mouse. The mouse on the far right is a mouse that has a inactivating mutation, the growth hormone receptor. Just as the humans were dwarfs, this mouse is smaller than a wild type mouse. And I'll just mention, and I'll come back to this in my talk, the mouse in the middle that's labeled GHA, that transgenically expresses an antagonist. It's an antagonist of the growth hormone receptor. It's similar to an FDA-approved drug that I'll talk about later in this talk. And it's intermediate in size. But basically, for the Lorone mouse, which is deficient for growth hormone receptor, it has a smaller body size. It has an increased lifespan that's statistically significant. That increase in lifespan hasn't been demonstrated for the humans that have this deficiency, but it's at least possible. They have very low rates of cancer and diabetes. It's also protected from genetically induced mammary tumors and genetically induced prostate tumors. And I'll just give you an example of the evidence that it's protected from prostate tumors in the upcoming slides. So I began working on this project through my collaborator, Steve Swanson, who's currently the dean of the School of Pharmacy, where I'm a faculty member. And he had been working on this years before I became involved in the project. And in the next few slides, I'll tell you about some of his work. So some of his work involved a model for cancer called the C31 SV40 T antigen transgenic model. So it expresses the SV40, which is a virus T antigen. This is a viral protein that inhibits the activity of P53 and RB. This is relevant for prostate cancer because human prostate cancers often have mutations in both P53 and RB. In this particular transgenic model, this antigen is expressed in the epithelium of both the prostate and the mammary gland. So in the males, these get prostate cancers with a defined timeline. And in females, they get mammary tumors, again, with a defined timeline. And the mammary tumors come up quicker. And as you'll see in my upcoming slides, that becomes relevant for how much this model is informative of the role of growth hormone in prostate cancer and breast cancer. But the work of my collaborator before I got involved in the project took the Lorone model, which is completely deficient in growth hormone receptor, basically from conception. So these are animals that never have growth hormone. And as shown in this panel here, these are smaller animals than a wild type animal. And this is the prostate from the wild type and from the Lorone animal. And you can see that the Lorone prostate is smaller. And these graphs quantify this. So overall, the body size is smaller for the Lorone mouse. And then VP and DLP refers to two different lobes of the prostate gland in the mouse. So the anatomy of the prostate gland is a little bit different in the human versus the mouse. And in the mouse, you have a ventral lobe, you have a dorsolateral lobe. And these are both smaller in the Lorone mouse. However, in this panel D, if you normalize the body size, the prostate is smaller, but it's proportional to body size. So it's not unusually small. It's smaller just as you'd expect with the decreased body size. And when they combine this with that SV40 driven model of prostate cancer that I just described, the difference when you were lacking growth hormone receptor was dramatic. So these upper panels are H&E stain slides, and they're at low resolution, so it's a little hard to see, but the purple density in these two panels on the left sort of indicates the buildup of prostate epithelial cells, which is the pin in this model that is largely absent in the panels on the right. And basically, the graphs at the bottom depict the incidence of pin, which is much lower in the Lorone mouse than in the wild type mouse, and the percent area of the prostate occupied by pin, which is again dramatically lower in the Lorone mouse. So this is similar to what was observed in humans. You have this genetic insult that should cause prostate cancer. If you don't have an intact growth hormone signaling pathway, you're basically blocking it. And this was work done before my lab got involved. But, you know, my question was, what if you can delete growth hormone receptor conditionally? Because in the clinic, if you're going to encounter a patient with prostate cancer, obviously, virtually all of those patients are going to have intact growth hormone signaling throughout their whole lives. And then the question really clinically becomes, what if you disrupt growth hormone signaling after they've already got cancer? And is that beneficial? And that's really the question we care about. So I'm going to tell you today about a couple different genetic approaches to doing this. One is going to be a global deletion of growth hormone receptor after lesions are present in the prostate. And the other is going to be specific deletion in prostate epithelial cells to see whether these impact the disease state. So in terms of the strategy for conditional deletion of growth hormone receptor, we're using a Cre-Lox approach. So we're using a genetic allele of the growth hormone receptor that has exon 4, which contains required elements of the protein surrounded by LOXP sites, and then that can be deleted by Cre recombinase. And I'll present data using two different genetic sources of Cre. One is a ROSA 26 mouse that expresses a mutant form of Cre called the Cre ERT2. So Cre ERT2 resides in the cytoplasm, is inactive unless you treat with the drug tamoxifen. When you treat with tamoxifen, that changes the conformation of that protein, allows it to enter the nucleus and be active as Cre, and then delete the gene of interest, which in this case is growth hormone receptor. So that allows us to control the timing of when growth hormone receptor is deleted. The other model we used is PB or probasin Cree. This is a version of Cree that's expressed specifically in prostate epithelial cells and not in other cells of the body. So you're asking what happens if you eliminate growth hormone signaling just in those prostate epithelial cells. So this slide is just a control slide that says, did we achieve the deletion of growth hormone receptor we are intending to? And so we're doing real-time PCR here for the transcript of the growth hormone receptor that includes exon 4. And in these experiments, we have a vehicle control, and we also have a tamoxifen control. And then we have those animals where we have the ROSA 26 Cree deletion. And we basically completely eliminate the growth hormone receptor transcript in the DLP, which is part of the prostate, and the liver when we use that ROSA 26 Cree. When we use the prostate specific PB Cree, we completely eliminate that transcript in the prostate and the DLP, but we don't eliminate it in the liver, which is exactly what we expect. And we do have a growth hormone antagonist model in this experiment as well, which is just expressing transgenically a molecule that's similar to a drug that inhibits the growth hormone receptor. And there are some effects on growth hormone receptor expression, but then also serum IGF-1 is dramatically lower with global deletion of growth hormone receptor, which we expect. And then the antagonist achieves a more modest reduction in serum IGF-1, but it does reduce serum IGF-1. So basically, this slide just shows you we're achieving the deletion of growth hormone receptor as we plan to. Now, one caveat for this experiment. So in this slide, I list the different genetic groups that we were targeting in this experiment. And unfortunately, and this is different than what's put out in the literature, these animals had a lot of off-site tumors, particularly head and neck tumors. And we hope to go to a fairly late time point because that's when most animals have invasive prostate cancer. But we were not able to achieve that because of these off-site tumors. So that's that's a limitation. It was unfortunate, but that's what happened, probably due to genetic background of the experiment that we did. So we looked at an earlier time point, and quite frankly, we worked with a pathologist. And as our pathologist looked at the prostates in these animals, there was less invasive cancer in PIN than we expected based on the literature. But again, that's what we got. And what we were able to do is to look at the pathology of the tumors at these earlier stages. And normally the prostate epithelium is a single monolayer of secretory cells. And we did get a piling up of cells which didn't meet the pathological criteria for PIN, but so we'll call it a stratified epithelium. And it had higher proliferation as indicated by Ki67 as a marker of proliferating cells in cell cycle. And when we looked at our different experimental groups, the global deletion or the prostate epithelial specific deletion of the growth hormone receptor reduced the amount of stratified area of the epithelium and reduced the Ki67 labeling index in that epithelium. This growth hormone antagonist expressing transgenic also reduced stratification and proliferation of the epithelium. So this is supportive of a role, but I'm not going to call this evidence that there's an effect in cancer because we just didn't see cancers that we expected based on the literature. I'm now going to diverge. So this is going to be the breast cancer mammary tumor part of the talk. So in this particular model, as I said, the males were reported in the literature to get prostate cancer, and we saw less prostate cancer than we expected based on the literature. However, the mammary tumors in this model occur at an earlier time point. So essentially 100% of invasive mammary tumors at 16 weeks in this model, which basically meant that early lethality we saw for the prostate tumors wasn't really as much an impact on the ability to study this. So this is again a slide that is the control showing that by treating with tamoxifen in the liver, we can delete growth hormone receptor and see complete deletion. We basically eliminate expression of IGF-1 in the liver when we do this. In the mammary tumors themselves, again, growth hormone receptor expression is eliminated when we treat with tamoxifen, and serum IGF-1 is dramatically lower. And so for the female study, what we did was, instead of particularly a particular time point, we monitored each female mouse, and for the experimental animals that were getting growth hormone deletion, as well as the controls, we began treatment when the largest tumor reached 200 millimeters cubed in size. So that was what was kept uniform in the study, is the largest tumor size. And when we did that and used tamoxifen to delete growth hormone receptor, what we found is for the two control groups, the volume change, and that's what's depicted here, the percent volume change of the tumors continued to go up for both control groups. And we actually got a modest regression of the mammary tumors in the animals with growth hormone receptor deletion, supporting the idea that these tumors require growth hormone signaling for their continued growth. We did analyze these animals for markers. Ki67 is a marker of cell proliferation, and we did see, after we deleted growth hormone receptor, a statistically significant decrease in proliferation. And the data for that are shown here, where in the non-growth hormone receptor deleted cells, almost all cells are in cell cycle, and it's dramatically reduced with growth hormone receptor deletion. We did look at cleaved inactivated caspase-3 as a marker of apoptosis. Our sample size at this point is relatively small. We did see higher cleaved caspase-3 in the growth hormone receptor deleted animals compared to the controls, but with our current sample size, the p-value was only 0.54, so not quite statistically significant. We suspect as we do more animals, that'll reach statistical significance. So we think it's both increased apoptosis as well as decreased proliferation that contributes to the smaller tumor size when we delete growth hormone receptor. Now, in terms of mammary tumors, we have looked at this pathway in more than one genetic model. And I'm going to talk briefly about a second model. So this is in rats. So there is a so-called spontaneous dwarf rat, or SDR, and it has a mutant inactive growth hormone gene. So the previous animals have growth hormone receptor mutant. This is the growth hormone itself. There's a mutation in one of the splice sites for the growth hormone that basically means these animals don't make active growth hormone. Again, this slide depicts some data from my collaborator, Steve Swanson, data collected before we were collaborating. But basically what his earlier data had shown was that these growth hormone deficient animals are resistant to MNU-induced mammary tumors. So you essentially get no incidence of mammary tumors treated with MNU if you are deficient for growth hormone. You can, however, supplement those animals with exogenous growth hormone, and then you can induce mammary tumors. And in the article that I cite at the bottom of this slide, that's what they did. And sure. I do not know the ER status. I know that in the mouse model of cancer that I described, it's known that, for example, it's resistant. It doesn't respond to, basically hormone ablation therapy. This rat, the MNU rat model, I'm not as familiar with. So I don't know. Yeah. Yeah. I did not check. I'm completely willing to believe that that's the case. But what we did as a follow-on study to those earlier studies is that we looked at, so that spontaneous dwarf model had been maintained on a very inbred background. And on that very inbred background, if you withdrew growth hormone, basically all of the tumors regressed, but it wasn't clear to what extent the genetic background played a role in that. And so in the study that we did, it was outbred to Sprague-Dawley rats to put it on a more outbred background. When we did that, I'm just giving you some examples of some tumor growth profiles, and the vertical dashed lines indicate when growth hormone was removed. And basically, the bottom line is many of the tumors continued to grow after growth hormone removal. So the growth hormone was required for the initiation of tumor development, but in many cases not required for continued tumor growth. And what we then did was ask, well, what is the sensitivity of these tumors to doxorubicin, a chemotherapy that's commonly used against breast cancer? And we used two different doses. 2.5 milligrams per kilogram would be a more normal dose for a rat model, and 1.25 would be a low dose. And when we treated these animals with doxorubicin for both doses, so what's shown here in the dashed line is the average growth, and the confidence intervals are shown in the gray, is that for the wild type animals, tumors continue to grow at both doses of doxorubicin. And for the animals where growth hormone had been withdrawn, tumors were basically killed off and shrank in response to doxorubicin at both doses in cases where there was no growth hormone signaling. And we looked at Ki-67 labeling index, and in the growth hormone deficient animals, it was dramatically lower. Again, these are for the animals at the lower dose of doxorubicin. Then we looked at tunnel. Again, the tunnel labeling index indicating apoptosis was much higher at that lower dose of doxorubicin for the growth hormone deficient animals. So sort of the idea here, and this is an idea that we have for both breast cancer and prostate cancer, is that we're not necessarily envisioning that targeting the growth hormone signaling pathway will be a monotherapy for cancer, but that our data support the idea that it can enhance the activity of chemotherapy to the point where you can potentially be more effective, but also you will use a lower dose where there's less side effects for a patient. That at least I'm more familiar with prostate cancer. A lot of prostate cancer patients, chemotherapy is extremely hard on them, and lowering the dose would be very beneficial. So I'm now going to transition from talking about genetic models to talking about more a pharmacological approach to investigating the role of the growth hormone IGF-1 axis in cancer. So there is a growth hormone antagonist drug called pegvisomant, and I show its structure here. Basically, it's a sequence and chemically modified version of growth hormone. So certain amino acids have been changed, and then it's been pegylated. And so those two things have two effects. One is they make it an antagonist rather than an agonist, and the pegylation gives it a longer circulating half-life. And this is a drug that's FDA approved for acromegaly, which is a condition where certain individuals have a hormonal condition where they make too much growth hormone, and it can't be surgically treated. I want to mention that there have been other publications in the literature looking at the role of growth hormone in prostate cancer, But one important caveat to think about when you look at those studies is there is a species specificity to the growth hormone action. The main thing being that human growth hormone receptor is activated by human or primate growth hormone, but it's not activated by bovine or mouse growth hormone. And one of the implications of this is that pretty much all of the common human prostate cancer cell lines have been immortalized in a condition that's free of growth hormone that can signal to their receptor. And also, when you do xenografting studies with human prostate cancer cell lines, unless you're supplementing with exogenous primate growth hormone, it's basically not seeing any growth hormone. And so one of the things that we did, we took into account this species specificity. So we've looked at prostate cancer cell lines that were derived from mouse models. So the first ones I'll tell you about are based on cell lines derived from the prostate-specific P10 deletion mouse. So there's 4 different cell lines like that that are referred to as P10P2, P10P8, P10Cap2, and P10Cap8. And the data in this slide are all growth in vitro, but basically all of these cell lines are grown in a standard media condition where they're seeing growth hormone that would be activating the receptor present on these cells. And all of these cell lines are growth inhibited by that drug pegvisomant, which I talked about as being a growth hormone receptor antagonist. We've done some work to ask, what signaling pathways might be responsible for this growth hormone dependent growth. And so these are immunoblots. We have clues from the literature what pathways might be activated. They include JAK-STAT signaling, ERK signaling, and AKT signaling. And basically what our studies suggest is that if we serum deprive these cell lines and treat with growth hormone, we get strong activation of JAK2 and STAT5. And so we think this is the main signaling pathway that is being utilized in these cells for growth hormone. Supportive of that, again, we're doing very similar in vitro growth studies to what I just showed you. And the chart over here on the right-hand side shows you what the different conditions correspond to for the different graphs. But this is, again, 4 different cell lines. The first line is just growth in the basal medium, not containing supplements or serum. If we add growth hormone, we get strongly induced growth. If we also include pegvisomant, the growth hormone receptor antagonist, we block that growth, which is, of course, completely expected. And if we add an inhibitor of the STAT3, STAT5 activity, we also basically block that growth, whereas a MEK inhibitor, UO126, does not block that growth. So these data support the idea that growth hormone is supporting the viability of prostate cancer cells via a JAK-STAT signaling pathway. Now, we've also taken these studies to an in vivo system where we've xenografted these cells. So just to orient you, and you're going to see several slides like this. So a site we like to graft cancer cells to is under the kidney capsule. It's a good site for cancer cells because there's a rich vascular supply. And so what you're seeing in this image, the dark red is the kidney, and the sort of more pale area is the tumor growing there. It's surrounded by the dashed line, and a scale is shown for just to give you a size orientation. But in this particular study, what we did was we took 2 different of those mouse prostate cancer cell lines. We established xenografts for 3 weeks. And then after 3 weeks, we began treatment with pegvisomant for 3 weeks. So it's 6 weeks total, 3 weeks of pegvisomant. And we saw a dramatic decrease in tumor growth when these were treated with pegvisomant. We analyzed the tumors for Ki67, which is a marker for cells in cell cycle or proliferation. And we saw a dramatic decrease in proliferation with pegvisomant treatment. We also looked at cleaved caspase 3 or active caspase 3 as a marker for apoptosis. And again, with pegvisomant treatment, we saw a dramatic increase in cleaved caspase 3, indicating higher apoptosis. We also took our xenograft tumors and we did RNA-seq to try to determine what genes are altered by pegvisomant treatment. And this is in this slide a volcano plot with genes that are increased on the right and decreased on the left. And basically, there were a large number of genes that were altered by pegvisomant treatment. Notably, we looked at the cosmic database for the cancer census genes. These are genes that are implicated as drivers of cancer. And 178 of the genes that were altered by pegvisomant treatment fall into these cancer census genes. And some are highlighted in blue on this slide. And we also found that there were basically, you could sort the genes that were altered by pegvisomant into two categories. So I mentioned the fact that growth hormone is secreted from the pituitary and it has two potential modes of action. One would be growth hormone acting directly on a tumor cell. And so this gene REM1 that I'm highlighting here falls into that category where when we treat a xenograft with pegvisomant, we have a dramatic suppression of this gene. If we take the same cells and we treat them in vitro in cell culture, we also get dramatic suppression of that gene. And then, as we might expect, if we treat those cells with growth hormone, we induce that gene. So we have the opposite effect of pegvisomant. And then IGF-1 has pretty much no effect. We saw a second category of genes illustrated by CDK4, where we have a dramatic suppression of this gene by pegvisomant in a xenograft. When we go in vitro with pegvisomant, we really don't see a statistically significant effect on this gene. And again, if we treat these cells in vitro with growth hormone, we're not seeing really a change in this gene. However, if we treat these cells in vitro with IGF-1, now we see an induction of the gene. So this implies this other category of gene, when we treat with pegvisomant, is indirectly regulated by growth hormone suppression via the endocrine effects, and in this case, in particular, by IGF-1. So, we think growth hormone for prostate cancer and the gene expression changes we're seeing is both having direct effects on cells, and we have some genes that are like that, and indirect endocrine effects on cells, and this is a gene that falls into that example. One area that is under development, and I'm not going to talk much about today, is just like in the mammary tumor studies, we're interested in the idea that inhibiting growth hormone may synergize with chemotherapy, which is an important form of therapy for prostate cancer. And so this slide is based on in vitro data where we treated cells with varying concentrations of pegvisomant and varying concentration of docetaxel, which is a chemotherapy drug commonly used in prostate cancer. And the formula in the upper right hand of the slide is the so-called combination index. When that value is below about 0.9, it's indicative of synergy for drug working together. And in this case, the compared to vehicle, both pegvisomant and docetaxel have some inhibitory effects on growth, but the combined effect clearly falls into the area of synergy, where you're getting synergy between inhibiting growth hormone and docetaxel. And this is for two different cell lines. So we haven't done that much in this area, but it's a future area of exploration for us where we think a potential future strategy will be that drugs like pegvisomant can be a synergistic add-on to chemotherapy for prostate cancer. Now, I do want to say that all the studies with pegvisomant I've told you so far have been with cells that have this PTEN driver mutation, and these observations are not limited to cells with that driver mutation. So in this slide, I'm showing data for primary human prostate epithelial cells, and we've shown that these express growth hormone receptor, and as we treat them with increasing doses of human growth hormone, their population expands faster, and you can inhibit that with pegvisomant. So, this is not something specific to the PTEN driver. We've also done similar studies with mouse prostate cancer cells derived from the TRAMP model. So this is a model that's driven by the SV40T antigen. So it's a different driver than the PTEN mutation, and overall, we see very similar results, which if we graft the TRAMP-derived prostate cancer cells into a mouse and we treat with pegvisomant, we see lower overall tumor mass. Again, this is establish a tumor for 3 weeks, then treat for 3 weeks. We see lower proliferation and increased apoptosis. And then the genes listed across the bottom of this slide, these are the same genes we identified by RNA-seq in the PTEN models. And although we don't see 100% correspondence in the gene expression changes that we saw in the PTEN model cells, in many cases, we basically recapitulate the gene expression changes we saw in the PTEN mutant cells. So that suggests to us that the findings we had in that PTEN mutant cell-based study are more generalizable to other models as well. Now, all of these data that led us to think about basically, does the growth hormone pathway act as a driver in prostate cancer in any way, rather than merely being permissive? And so this is an analysis of data from the Cancer Genome Atlas. And what it basically shows is that for the growth hormone receptor, there's statistically significant overexpression of the growth hormone receptor in prostate cancers that have ERG fusion genes or ETB1 fusion genes. And collectively, this is about 50% of prostate cancer cases. And obviously, when you see a gene expression change, it can be a driver of cancer, or it can be a passenger. And one reason that gave us hope that this might be a driver change is another study, and I don't know if anyone in the room is in the groups that use transposon-mediated immunogenesis in the mouse to study cancer. I know certainly that was pioneered here at Minnesota. But as I looked through so there's not evidence from prostate cancer that growth hormone receptor could be a driver change. But as I looked through the literature, there was this study of leukemia using transposon-based technology to look for drivers of leukemia. And basically, what I've highlighted here is that both ERG mutations and growth hormone receptor mutations were driver mutations as activating mutations in leukemia. And there was co-occurrence of ERG and growth hormone receptor activating mutations as drivers in this leukemia study. That gave me hope that maybe this overexpression of growth hormone receptor in prostate cancer might reflect a driver activity of growth hormone receptor. So we went ahead and we established some cell line models where we overexpressed growth hormone receptor in some of these mouse prostate cancer cell lines. So the upper graph here, this is expression on the right, or so the left-hand side in the PTEN-P2 cells and TRAMP-C2 cells. So one of these cell lines has the PTEN driver, one has the TRAMP driver. In both cases, we're getting roughly threefold overexpression of growth hormone receptor. And In both cases, under normal media conditions, this leads to cells that grow faster in vitro if they overexpress the growth hormone receptor. And then we did xenografting studies as well. And for both overexpressing cell lines, we get dramatically more higher tumor growth for the tumors overexpressing growth hormone. And this corresponds to xenografts with a higher Ki67 labeling index, which indicates higher proliferation. So we do think that potentially growth hormone receptor overexpression is a driver change in prostate cancer. And we also went on to look at, these are all genes that we found were influenced by pegvisomant, so inhibiting growth hormone receptor expression. And in each of these cases, genes that were altered by pegvisomant in the xenograft context had the opposite change when we overexpress growth hormone receptors. So it's basically what you'd expect is if you hit the growth hormone receptor antagonist, for example, these genes go down. If you overexpress growth hormone receptor, these genes go up. So we do think that overexpression of growth hormone receptor in a subset of human prostate cancer cases is likely a driving change that contributes to cancer progression. So to summarize, what I told you about today is conditional activation of growth hormone receptor reduces epithelial proliferation in the C3-1 SV40 tangent transgenic model. It also reduces growth of mammary tumors in that same model. Pegvisomant, this growth hormone receptor antagonist drug, inhibits mouse prostate cancer growth in vitro and in vivo. And I described for you several gene expression changes of cancer relevant genes. And also that growth hormone receptor overexpression is present in some prostate cancer cases in humans. And based on our evidence from modeling that in mice, we think that that could be a driver change that's driving prostate cancer growth. And ultimately, of course, what we think is that drugs like pegvisomant might be useful as prostate cancer therapies, potentially in combination with chemotherapy. There's been a lot of clinical studies using this type of drug for acromegaly, which is in people that have overexpression of growth hormone. There's been very little clinical study in oncology as a whole. And we think such studies would be potentially very interesting. So in terms of thanking people, Chris Unterberger is a graduate student who recently defended his PhD thesis in my lab. He's the driving force behind a lot of the data I showed today. Valena is a research specialist who contributed a lot. There are a lot of other people listed on this slide that contributed. My key collaborators especially include Steve Swanson on the growth hormone project, and Rong Hu is a pathologist who works with us on prostate. Stephanie McGregor is a pathologist who works with us on the mammary tumors. I have key collaborators in benign urology as well. I didn't really talk about any of that research today, but I'm happy to take any of your questions.
So I have the mic. Do you see any developmental differences in the mammary gland or prostate in the first part of your studies, or stem cell number differences?
Yeah, so that's a really interesting question, and I've encouraged my students to look in great detail at that. I think that the easiest answer is both the prostate and the mammary glands are smaller in the growth hormone receptor deficient animals, but everything that we've seen so far says they're proportionally smaller, meaning proportionally fewer branch points, proportionally, just smaller. But like nothing stands out as unique to growth hormone.
Yeah, so I noticed when you inhibited GH that AR expression was going up,
Yep.
or if you overexpressed it, it was going down. Have you ever tried looking for synergy with AR inhibition with like enzalutamide, abiraterone, things like that?
Yeah, so that's a really good question. So other investigators have published studies that have used pegvisomant with those AR antagonist drugs. One of the caveats of their studies is that they were looking at human cells grown in a mouse, and they didn't supplement the mouse with growth hormone, like human growth hormone or primate growth hormone. So there's data. I would say the studies were not the ideal studies because the cells they were using, they could see the indirect effects of growth hormone like through IGF-1, but they could not be directly activated by growth hormone because they didn't supplement with growth hormone. So there's some data out there. I would characterize it as incomplete.
After you.
A question and a comment. So the question is the pegvisomant chemotherapy story. So most people think that, the dogma is that less proliferative cells are less sensitive to cytotoxic chemotherapy because they're not going through cell cycle. But you see synergy. So how are you thinking about that?
I mean, I guess my thought is that the targets of growth hormone signaling are a lot more diverse than just proliferation. So, if you look at growth hormone activity in general, it's regulating many aspects of metabolism, right? So, in those Lorone patients, they're deficient, they have less diabetes. When people do tissue-specific deletion of growth hormone receptor, you affect fat metabolism, you affect a lot of different energy metabolism. And so I get what you're saying about the proliferation being low. If it's less proliferative, you'd be less sensitive to chemotherapy. But I think that not having active growth hormone affects the cell in a much more profound way than just lowering proliferation. And I do think also there's two different things that have been documented, not by my lab, but by other labs in the literature that relate to this. One is that in certain tissues, inhibiting growth hormone signaling does downregulate some of the transporters that basically are pumps that pump chemotherapy out of cells. And, other mechanisms of apoptosis resistance are downregulated when you get rid of growth hormone. So,
I agree with you. It's probably something else like survival. So the comment I have, because I just looked it up, if you have a source of pegvisomant, we should talk about it. I submitted a phase 1 trial for pegvisomant and doxorubicin in 2005. That's the part I had to look at. And they said no. Pfizer, who owns the drug, it's a hard drug to make. And they also really just said they didn't think it had a role in the cancer market. So if you can find somebody that can make you pegvisomant.
So I did get my drug.
I got a phase 1 trial written.
So I did get pegvisomant from Pfizer. Probably I've done all the experiments that I technically have agreed to do with them, but I still have some left. I'm supposed to destroy it, but I haven't destroyed it. But I definitely don't have enough to do like a human trial.
So that's really cool, Paul, because I feel like, if you're talking about metabolism, you might be killing G0 cells. That's why you might get better kill if you're inhibiting growth and G0 cells, which are going to be cells that would be supporting a stem cell compartment. So even though the tissues are smaller proportionately, they might have disproportionately smaller stem cells, right?
Yeah, no,
they're easy to quantify, right? So that would be really cool.
And
if you're doing that.
We have not tried to quantify stem cells in particular.
Paul, this is terrific. Always have to introduce the word metastasis in this whole thing. is there an impact when you are interfering with growth hormone receptor? You affect the growth. Oh, use the mic. How's that?
Yeah.
Did you hear what I just asked?
I did
You
hear.
but nobody else
You're,
did.
you're asking about
So,
metastasis.
yeah, so the question is there an impact on metastasis? Because cells, when they're not proliferating, can certainly be invading.
Yeah.
Going to other
And
places.
so the cell line models that I presented today are not really known to be metastatic. What I did not present today is that we have shown that, for example, pegvisomant in vitro growth inhibits the 4T1 mammary tumor cell, which is a cell that does go metastatic. And we were very interested in the question you just asked. And we like to graft cancer cells under the kidney capsule because it's a great place for cell survival. But it turns out those 4T1 mammary tumor cells are so aggressive and fast growing that when we grafted them under the kidney capsule, like it obliterated the mice almost immediately. So like basically, no, I mean, so basically we tried to do an analogous study to what I showed with the 4T1 mammary tumor cell. And it was unsuccessful, not because, but because of technical details, because basically we tried to go 3 weeks of establishing the tumors and the mice were just obliterated. And so I think that we could do a study with those mammary tumor cells that are sensitive to pegvisomant, do go metastatic, but we have to back off on how we put those cells into mice because they're so aggressive. It's beyond what we normally experience with the prostate cells.
I mean, a related question is, I'm sorry, do they impact the growth of the metastasis once it's formed?
Sure, and those are great. Those are great questions, and I think that you know, a really good experiment that we haven't done that we could do would be to you know, put those mammary tumor cells into a mouse. Maybe we even pre-treat with pegvisomant, or we put less cells in, or, you know, there's a number of ways you could do that experiment that would be more successful than what we actually did.
All right. Well, thank you. Oh, we have one more question.
In the beginning, you were talking about off-target increases in head and neck tumors in the cream-mediated conditional inactivation. What kind of head and neck tumors were you getting?
Yeah, so it was actually probably not due to that CRE, but due to the C31 T antigen model. And, you know, so number one, and I don't know if this is an issue here, but some of the best animal pathologists at Wisconsin have left for private industry over the last several years. So we actually have a real problem at Wisconsin with getting support for general mouse pathology. I've had to work really hard to find some relatively junior faculty to do the prostate and breast cancer pathology work with me. That they're great. I love them. But the long answer is that we think it's, we sometimes see them on the neck. We think there's some nasal passage tumors. We haven't had complete pathology analysis from that. And our general animal pathology core has actually told us we're so understaffed now that like we just can't do it anymore. And so that's a problem at Wisconsin. We had a great pathologist that worked with us named Ruth Sullivan, and like maybe 4 or 5 years ago, she got recruited away to Genentech, and like Genentech's letting her do it essentially all remote. So like she can sit in her house with her high resolution pathology, and yeah, she's a friend, but still it's hard. could be lymphomas as well. Yeah.
All right. Do we have any more questions? If not, let's say thank you one more time to Dr. Marker for